second keynote presentation this morning. I am Dr. John Schooner. For those of you who don't know who I am, I'm an associate professor of kinesiology at Oregon State University. I'm also the current president-elect and soon to be president of ACSM Northwest. Before we begin the presentation, I want to cover some of the etiquette which we've done for our previous presentations. Please ensure that you have your microphones and cameras muted throughout the presentation. If you have any questions about the presentation, send those to me directly via the chat function. I will then relay those back to our presenter once we reach the Q&A portion at the end of the presentation. All right, with that, it is my pleasure to introduce our final keynote speaker for our virtual 2021 annual meeting, Dr. Stephen Heymsfield. Dr. Heymsfield is a world-renowned expert in the area of human body composition and human body composition assessment. Dr. Heymsfield originally obtained his MD at Mount Sinai School of Medicine in New York in 1971. He has since held faculty appointments at Emory University in Atlanta, Georgia, and Columbia University in New York. Dr. Heymsfield also has significant experience working in the private sector, where he previously served as Global Director of Scientific Affairs related to obesity at Merck Pharmaceuticals. He is currently a faculty member, professor, and director of the Metabolism and Body Composition Laboratory at Pennington Biomedical Research Center, which is a campus of the Louisiana State University System in Baton Rouge, Louisiana. Prior to his current position, Dr. Heymsfield previously served as the executive director of the Pennington Biomedical Research Center as well. In terms of research, Dr. Heymsfield has authored or co-authored more than 650 peer-reviewed journal publications, half a dozen books, and over 30 book chapters. He is a fellow of the American Society of Nutrition and the Obesity Society, and currently serves on the editorial boards for the journal Obesity, and previously served on editorial boards for the American Journal of Human Biology, American Journal of Clinical Nutrition, and the International Journal of Body Composition Research. And just recently, he served as an advisory committee member for the development of the USDA's 2020 dietary guidelines for Americans. All right, and with that, I will turn it over to Dr. Heymsfield for his presentation entitled Advances in Body Composition Research.
John, thanks very much. I really appreciate the invitation. And I'm going to start off by disclosing that I'm in the medical advisory boards of Tanita Corporation and Medifast. Well, this topic is of great interest to me, this idea that we're moving away from broad generalities about people in general physiology and down to individuals. And if you think about an individual person, what determines their body size and shape and composition, we know that there are multiple independent variables like height, adiposity, age, race, genes, activity levels, of course, hormones, drugs, and so on. So there are a lot of variables that affect our body composition. And taken collectively, each of us has a unique body composition, not different than a fingerprint or a retinal scan. Each of us is different. And those differences are really the theme of my talk. And I'll end with a note about movements in that direction to understand individual differences. And if we think about a person's shape, that in turn is related to what they're made out of, their body composition. And in turn, their body composition links up to many factors, as you're well aware of, such as health risks, athletic performance, energy expenditure, metabolic effects. I'll talk a little bit about that as well. And this is a roadmap for our presentation today, and particularly how shape ultimately relates to body composition and in turn to various outcome measures. And today, our approach to this, as you know, is extremely simple. For most, certainly in the clinical setting, we measure weight and height, and I don't think they're measured particularly well in most clinical settings, but we measure those two. We use them to calculate body mass index. And in some places, you can get your waist circumference measured or perhaps your hip, but that's about it. And what I'll show is that there's a real transformation taking place that your field in particular is part of. So over the next 40 minutes or so, I'll talk about some of these advances, and I'll really begin that process by giving a bit of a historical overview. And if you want to reach me, that's my email address there. It's my name at pbrc.edu. So, some of the earliest work began with an interest in physical efficiency in soldiers' work capacity. Matiega, a great Czech uh, anthropologist, he was really one of the first to try and put together a format for body composition. And his idea was quite simple, and he used various uh, anthropological tools to make this assessment. And he thought about osseous or bone, dermal and subcutaneous adipose tissue, muscle, and the remainder of the body. And that concept then has permeated down several generations, including my own generation, in terms of partitioning the body into these different compartments, going from the shape of the person measured by various calipers and devices like that, down to these major body components. That was 1921, exactly 100 years ago. Remarkable because you'll see the transformation that's taken place over 1 century. And then a great era began right at the cusp of World War II, late 1930s up until the mid 1940s. And some of this work involved, for example, X-rays. Röntgen discovered X-rays are late 1800s. And by 1940, Stewart, who was at Harvard, used X-rays in adults and children. And what he would do is he would measure the soft tissue diameters in these X-rays as a measure of muscle and fat mass. And this became quite popular. It's hard to imagine doing X-rays today with the radiation exposure, but this is how they did it, 1940. And then another great milestone, very influential in your field, Benke, who was in the Navy studying things like the bends, developed what we now recognize today as beginning with the underwater weighing technique and now air displacement plethysmography. But Benke's idea was simple. He thought of the body as two compartments, fat and fat-free mass, the remainder, each with a known and stable density. The density of fat being zero point 0.9 kilograms per liter and the density of fat-free mass being 1.1 kilograms per liter. And then if you put someone in a tank with water and you weigh them underwater when they exhale, and then you weigh them in air, that gives you their body density or volume underwater, and then weight in air. Uh, that gives you their body density. And from body density, Spenke wrote out a series of equations, and later workers contribute to this, like William Seary. And you could then solve for the mass of body fat by putting in the volume you measure in the tank and the weight in air. So that was Benke's method. And then another technique developed, starting with Carl Dusek, was ultrasound. He worked on sonar, again, also in the Navy. And Dusek worked on these sonar devices in around 1942. And he had very primitive methods. He used a water tank to create homogeneity, and using primitive ultrasound devices, certainly primitive by today's standards, he could quantify, for example, the structure of a limb, showing very crudely the outline of the limb, the muscle and adipose tissue compartments. These were the earliest studies of body composition during that time. And then right at the end of the war, 1945, Nello Pace and Edith Rathbun wrote a seminal paper, very fascinating discovery. They also considered the body as two compartments, fat and fat-free mass. And all of the water in the body is in the fat-free mass compartment. And what they discovered across, just shown in this little graph here on the top, is when they plotted the slope, or they looked at the slope of the line of total body water versus fat-free mass going from mouse basically up to humans and very large animals, they found the slope was constant at 0.73. That is 73% of fat-free mass is water. And the reason that's important is because we have three isotopes of water, tritium, deuterium, and oxygen 18, that can be used to measure total body water. And once you know total body water, then you use that constant 0.73, and it gives you a derivation of fat-free mass. And then once you know fat-free mass, you simply subtract that from body weight and you get fat. So hugely important technique in our field. And these isotopes are all readily available. Deuterium is the most common in use because it's relatively cheap and not radioactive. Tritium, not so much anymore today because of the radioactivity. And oxygen 18 is rather costly. We don't use that too often. But body water is easily measured and it's a very important technique. And here's just some of the equations you use for this approach, but they're extremely simple. Probably the next major jump in body composition work occurred in the 1970s, now 20 years later or so, with the introduction by Godfrey Heymsfield of computerized tomography. That's Godfrey there with the first head scanner for CT. And it was very soon thereafter that CT became widely available. And it was found that with a CT scan, for example, shown here, you could get a cross section of the human limb, a very high resolution cross section. And that cross section was far better than those radiographs by Stewart 20 or 30 years earlier. And you could take the data in the image and you could plot a histogram, pixels, these are called picture elements. And the picture elements fall out into two curves, nice curves you can see. One is the adipose tissue curve and the other is the muscle tissue curve. And that allows you to quantify the area of muscle and the area of adipose tissue in each slice. And you can basically add up the whole slice. The other wonderful thing about CT is for the first time you could see literally inside the abdomen. And this just shows the visceral adipose tissue lit up in white here. And you could reconstruct organs. This is just a very early mock-up of slices through the liver, 1 centimeter apart. You put the pieces together mathematically and you get liver volume. This is a great advance because it was the first time this could be done. The other thing is you could get the density, radiographic density of organs such as the liver. And the liver might be perceived as two compartments, lean liver tissue and fatty liver. And that density then can be used. This is, again, very early demonstration of it. But if you plot the percentage of liver fat against its attenuation value, now known as the Heymsfield unit, then you could get the proportion of the liver as fat. And this technique is still widely used today to measure liver fat with CT. Just a footnote that the CT number, Heymsfield unit of water, is 0. And if it's a plus measurement, then it's lean. And if it's a minus measurement, it's adipose tissue. So we use those numbers to separate the tissues. Then also emerging after World War II, and then up in certainly most prolifically during the 1980s, Stanton Cohn, who worked at Brookhaven National Laboratory, pioneered a technique called neutron activation analysis, in which you take a source, for example, plutonium or beryllium, and you irradiate the subject briefly, and then you detect the various gamma rays and other rays that emerge after that brief irradiation. And it sounds a little questionable today when we're concerned about radiation exposure, but this is a miraculous technique. This is just an example. This is subject going into the irradiation chamber. The plutonium is drawn up through these tubes, briefly radiated, and then rushed over to a whole body counter. This counter then counts the decay. And from that, and other devices like this, we can get total body carbon, nitrogen, hydrogen, oxygen, calcium, phosphorus, sodium, and chloride. And as you can imagine, once those components are all known, you can basically do a chemical analysis of a human in vivo. And so this technique was extremely influential in teaching us about human body composition at a very high detail level. But those centers have largely disappeared today because of the radiation exposure and the very high level of expertise needed to operate them. So with this approach, it's really the only accurate approach for measuring total body protein. You can divide that into muscle and non-muscle protein, bone, water, and fat. So this is a wonderful technique, but it's largely gone today. And part of the reason it's been largely replaced by made in the early 1970s by Richard Mazes and his colleagues, where he found that he was looking for ways of measuring bone density and bone mass. He's an anthropologist. And at the time, they measured bone at the wrist because the wrist is almost all bone. If you feel your wrist, you'll see there's very little soft tissue there. But when you get to other parts of the body, like the hip, which he was very interested in because of fractures, there's a lot of soft tissue over that. And so that required a technique, a dual energy technique, to separate out fat from bone in those regions. And he figured out a way of doing this with an isotope, gadolinium, that has two emitted energies. And this just shows Dr. Mazes with his early calibration instruments. And here's his first scan using gadolinium. He did a whole body scan, and you can see, and from that, he was able to separate out three components: fat, lean soft tissue, and bone minerals. And here's his results of his first scan. And then it wasn't too long until they replaced the gadolinium with X-rays. By the 1990s, this technique then permitted two energies instead of the isotope, two energies from X-rays. Now it's called, of course, dual energy X-ray absorptiometry, or DEXA. And DEXA then made these nice clean images of the body. And today we have DEXA scanners that you can get a morbidly obese person into using a very large scanner. Most scanners won't accommodate somebody this size. But as you know, we have at least two manufacturers, GE and Hologic, that make these DEXA scanners. And our kinesiology department has its own DEXA scanner, and DEXA provides a remarkable number of very valuable measurements. And I'll just briefly mention them. One, of course, is bone. And until DEXA, we couldn't measure bone any other way except with neutron activation analysis. And today we can measure bone easily. And the other thing that DEXA did for us, and DPA, dual photon absorptiometry, did for us, it allowed us to take Benke's underwater weighing two-compartment model to four compartments. And the way this would work then is, instead of just fat and fat-free mass as the original two-compartment approach, if you also measure total body water with deuterium and mineral or bone mineral with DEXA, then you can divide the body into four compartments: fat, total body water, bone mineral, and residual mass. And most of the residual mass is protein. There's a little bit of glycogen. And this became a very valuable technique. It's the way we calibrate many of our different methods, because this approach has the least number of assumptions in it. We don't assume hydration of fat-free mass is 0.73. We don't assume the density of fat-free mass is 1.1. We account for each of the individual components. And so it's a very valuable approach. And this just gives you some of the more or less density values that are assumed in that model and some of the typical equations. And when these were first introduced in the 1980s, these multi-component models, underwater weighing was the technique that was used to get whole body density and body volume. And that, of course, today has been replaced by air displacement plethysmography. And there's really only one system available now. There used to be many systems available, but today we use the BOD-POD system to get body volume. So if you then measure body volume with a BOD-POD, bone mineral with DEXA, and total body water with, say, deuterium, you can then get this high-level reference multi-component approach. Now, another very major advance took place. See, one of the problems with CT is that it has a fair amount of radiation that comes with it. The wonderful advance came with magnetic resonance imaging. With CT, there was one Nobel Prize. Godfrey Heymsfield got a Nobel Prize for CT. And then since then, magnetic resonance imaging has also come into play where you can get high quality cross-sectional images. And there have been 7 Nobel Prizes so far. There may be more. I expect there'll be more for the development of nuclear magnetic resonance and MRI. And this is just a little portion of the paper by Lauterbur, which he got his Nobel Prize for, and where he showed that you could mathematically reconstruct an image from an NMR signal. And I always love showing these kinds of papers. I write these long papers, take me months. This paper is less than 1 page long, and he got a Nobel Prize. So I'm working in that direction. That's a hard one, as you know. And then scientist named Demadian, a very interesting professor at Downstate University in New York, first patented this bizarre looking device in the 1970s, late or mid 1970s, based on these discoveries. Here's Dr. Demadian getting into his device. And ultimately, this was the first real MRI scanner. And today, of course, we have modern devices, but Selberg in 1992 first used it for body composition research, and this is, of course, a modern scanner today, and that's been transformational. There's many of you may know Bob Ross, who's from Canada. Bob introduced a whole body MRI technique in which basically you slice a person up head to toe, and then you get all these cross-sectional images. Then you hand segment each image to quantify the amount of adipose tissue, skeletal muscle, visceral organ mass, and these various components. It's a very expensive and tedious process. It takes at least a day to read a whole body scan and perhaps an hour to do the image itself. But Bob's technique then allowed us to essentially put all these slices together and get whole body muscle mass and adipose tissue. It's a tremendous advance in body composition research. And then it wasn't long after that, a scientist named Dixon developed a technique. Instead of the slicing up the body that way and hand painting each compartment, he developed a technique called water fat imaging. You use 2 different sequences, and it allows you to create a water image and a lipid image. And you can subtract those 2 images from each other and quantify directly the amount of fat and the amount of lean tissue in each image. And that can be automated. And this just shows 2014, a while back, but these automation procedures are advancing very rapidly, and you can semi-automate the analysis process. And this just shows that you can get individual skeletal muscles, individual fat depots, and so on using this semi-automated approach. Of course, you have to pay for it. It's not free, but nevertheless, the amount of time it takes to hand read one of these images is substantial. And I promise it won't be long before these automated techniques become much more sophisticated, using artificial intelligence and much more available to us. And I should say a whole body MRI Dixon scan only takes about 10 minutes, very short, not very long. Now, another major advance was Zimian Wang, great Chinese scientist. He developed the idea of the five-level model of body composition, in which the body is divided into atomic, molecular, cellular, organ tissue, and whole body levels. And the reason this is so important is that each level is distinct. And if you sum up all of the components in each level, it equals body weight. And the idea of this is so relevant because then you can make mathematical models at each level that solve for all of these various components. And as you saw in the 1940s, Benke with his underwater weighing technique, he simply had fat and fat-free mass, just shown here. That's a molecular level compartment. That was the simple compartment we had back then. But today, we can measure all of these compartments and many more of the body that are shown in this slide using these various mathematical models to solve for the different body components. And another major advance, and still has huge potential to go further, is Wei Shen around 2003, working at Columbia. This just shows the uh, reference man. This was a human subject who donated his body and was sliced into 1 millimeter slices. And you can see all the different adipose tissue compartments of this person. There's also a woman. And what it reveals to us in great detail is that fat or adipose tissue, much more specifically, it's not one compartment, it's many compartments. And we're only now fully appreciating the biology of these different compartments. And you can see this bottom right panel, there's a little different distinction in the MRI scan or CT scan, whichever that is, showing different types of subcutaneous adipose tissue based on density. But basically, we know there's epicardial fat, perirenal fat, visceral fat, and so on. And we still haven't completely defined the taxonomy of these different fat compartments because anatomists over the centuries were not interested in adipose tissue at all. They were interested in the visceral organs and muscle and other components, and they threw away the adipose tissue. And now we're discovering great roles for these adipose tissues, like bone marrow adipose tissue. There's a whole biology surrounding it, and there's still an opportunity to name and discover these adipose tissue depots. The other thing that imaging technology really brought forward in a major way for us is that we could reconstruct the organs in the body. You can quantify heart mass, brain mass, liver mass, kidney mass, spleen mass, and in addition, of course, total body muscle mass, adipose tissue, and so on. And what we know from in vitro and very few physiological experiments in living people, how much energy each of these organs consumes. So for example, the brain consumes about 240 calories per kilogram per day. If you have a brain roughly 1.5 kilograms, you can see that it's roughly 300 calories, or 15 to 20% of our energy expenditure comes from brain at rest. Liver is a high metabolic rate organ, 200 kcals per kilogram. Bone is very low. Muscle is actually quite low, 13 kcals per kilogram per day. Only second lowest to adipose tissue, which is 5 kcals per kilogram per day. And then we have organs such as the heart and kidney, which have extremely high metabolic rates. Heart's 440 kcals per kilogram per day. most of us have a heart of around 300 grams. So in the end, heart is only about 10% of our resting metabolic rate, very, very high. And so we're really heterogeneous. And these imaging techniques like CT and MRI have thus enabled us to quantify the great heterogeneity in body composition at this organ tissue level. And from, from that data, then, you can estimate someone's energy expenditure if you measure all of these components and then use these so-called constant, stable constant values, then you can estimate their resting metabolic rate. If you think about body mass is basically the sum of all our organs and tissues. And likewise, our resting metabolic rate is the sum of all of the organ and tissue resting energy expenditures. So if you go through a scanner, you get all these measurements, we can calculate your resting energy expenditure then and each of the components. And here's a little slide that gives you a sense of how all these parts connect then. Body composition and energy expenditure. If you think about, we eat food and we consume oxygen, and that's at the whole body level of body composition. And then when we go from there, each of these organs then utilize those nutrients and oxygen to burn energy at the rates I showed in the previous slide. And that occurs at the cellular level, of course, in mitochondria. And ultimately, we produce heat and CO2 and water and urinary urea and nitrogen as the end products. And we can therefore think of energy expenditure associations with body composition along these complex pathways rather than our very simplistic approach typically used, like let me know your weight, height, and age and sex, and I can tell you your resting metabolic rate. That's a very primitive technique that we use today, but I'll show a little bit more about that in a second. And here's another way of thinking about it. Here's our heat being produced on the right. And here are the various phenotypes. And let's make believe that all of these people have the same exact weight, body mass, and same age. Well, we can vary age a little bit, but we might have someone who's obese and has a high adipose tissue mass, but someone at the same weight who's highly muscular and has low adipose tissue mass. If you think about the differing specific metabolic rates of adipose tissue and muscle, of course, you realize that the muscular person for the same weight and height and age will have a higher metabolic rate. And you can show that very nicely using these various models. And you can stretch people out, you can keep their weight constant and increase their height. And what you see is their organ proportions change. For example, as people get taller, their brains don't get much larger. And as a result, the brain, which is a very large part of resting metabolic rate, becomes a smaller and smaller percentage of your metabolic rate as you get taller. And those then phenotypic characteristics then translate to various metabolic activities like protein synthesis. And those occur in the various organs and tissues, just showing the whole body here. And then as we move along the right here, we get simpler and simpler models. For example, at the organ tissue level, we have adipose tissue and adipose tissue free mass, adipose tissue free mass reflecting all these different organs and tissues. And then very widely used model, fat and fat-free mass at the so-called molecular level. We often have prediction equations for energy expenditure based on fat and fat-free mass, but reflecting this myriad of processes and organs and tissues underlying all of that. And we're getting better and better at quantifying these different contributors to resting energy expenditure. And just to carry this idea one step further, then, if we think about energy balance as the difference between how much food we take in and as energy and how much waste product we put out as CO2, urea, and heat, we can measure the differences between those two, energy intake and energy output, in a metabolic chamber, either a direct chamber or an indirect chamber, direct being heat production, indirect being CO2 and urea production. But we can measure all of those. And if we put someone in a chamber and we measure food intake and we measure the exhaust products, then we can calculate the change over 24 hours in fat, carbohydrate is glycogen, protein, and water. This is the most sensitive body composition device we have. We can measure these extremely small changes. And if we get balances of calcium, potassium, and sodium by collecting urine and knowing what went in, we can calculate changes in bone, protoplasm, and extracellular fluid. So these are not widely available, but these are the most sensitive body composition devices we have. Now, so far, I've really talked about mass, big organs like heart, liver, brain, muscle tissue, and so on. But of course, the big advance of the last decade has been looking at quality of tissues. And I'm going to give two examples. One, of course, is that adipose tissue and lipids are found not only subcutaneous compartment, but throughout the body within organs. For example, of course, liver hepatocytes have lipid in them, and that's what causes fatty liver or non-alcoholic steatohepatitis and other problems like that. But we can quantify that with MRI. So not only can we measure the total mass of the liver, but we can measure the proportion of the liver as fat. And likewise, we can do the same for pancreas, kidney, heart. We can measure the fat and adipose tissue contributions to all of these organs quite easily now in vivo. And we can also segment out, as shown in this panel here in pink, the various visceral adipose tissue compartments, the mesenteric, the omental adipose tissue. And likewise, we can get the intermuscular adipose tissue that's just shown the legs of a relatively healthy middle-aged person, a remarkable amount of adipose tissue. This is the marbling that occurs between the muscles. And this intermuscular adipose tissue correlates reasonably strongly with things like insulin resistance, strength measures, and so on. And finally, it's hard to ignore what's become very quite popular topic of discussion today, various types of fat cells, white fat, the beige intermediate fat, and the brown, high energy brown fat found in the clavicular areas and so on. All of these can be quantified. And so now we're going from whole body compartments down to detailed quality of tissues. Now, another place this becomes very relevant is muscle tissue. We often think, at least certainly I did when I began this work, as muscle as one large major body compartment. But this reveals that it's not. And this slide shows what happens to muscle mass in blue as you get older. Of course, it decreases, but muscle strength, shown in orange, decreases much more than muscle mass. Well, you're the exercise physiologists, but the first explanation, of course, maybe there's a training effect, a molecular effect, and so on. But there's actually more than that. And that is that as we get older and we lose muscle mass, we don't lose as much as you would think because muscle composition changes. And here's an example of, this is an extreme example in patients who had pancreatic ductal carcinoma and lost body weight. And it just shows on the left the sort of normal muscle tissue and non-cancer controls and the changes that occur as you progressively lose weight in the muscle tissue. And if you go all the way to the right, you see there's a lot of fatty infiltration in the muscle. And also this blue stain is connective tissue. And what you have, not only is you lose weight with cancer, but as you get older, and your muscle decreases in mass, the proportion of non-contractile elements increases. These little adipocytes and the connective tissue form a larger and larger percentage. And what happens with techniques like DEXA? DEXA is probably the most widely used approach for quantifying total body muscle mass today. And the way that technique works is that with DEXA, which, as you probably know, very easily accessible technique, you can quantify the lean soft tissue mass of the extremities. And from that, you can build a model. And this appendicular lean soft tissue then of the extremities has a very high proportion of muscle in it. And that then allows you to build a model that gives you appendicular skeletal muscle. And then if you look up, a number of studies shown appendicular lean soft tissue comprises a very large fraction of total body muscle mass. If you then get a DEXA scan, you measure appendicular lean soft tissue, you can use a prediction equation for total body muscle mass from that. And this approach is widely used in studying sarcopenia in particular. But what people find is that it's not quite that great for predicting strength and outcomes. And there's a reason for that. And I'm about to show that to you. But another approach, which will take us in that direction, is that almost 100 years ago, a number of investigators found that creatinine in the urine was highly correlated with total body muscle mass. And they develop a little equation. I'll just briefly describe the physiology that almost all of body creatine is in muscle. It's a little tiny bit outside of muscle. And that creatine breaks down like a clock at a constant rate and is excreted in the urine, such that for every gram of creatinine in the urine, that's equal to 22 kilograms of skeletal muscle. So if you collect a 24-hour urine, be careful how much meat you eat, collect a 24-hour urine, measure the creatinine, multiply by 22, that gives you your muscle mass. And Wang in 1996 and others since then have shown that that approach works reasonably accurately if you can get accurate urine collections to predict muscle mass based on either CT or MRI. Now, the reason I bring this up is because that technique has given way to a newer approach called deuterated creatine. You can take a little dose of deuterated creatine, a stable isotope. It distributes in your total body creatine pool, and then you get a urine sample a day or two later, and it gives you total body creatine, which in turn, using a constant again, can be converted to muscle mass. It's a very interesting technique, and Bill Evans and others worked to develop this. But what's fascinating about this technique is that it measures muscle cell mass as opposed to DEXA, which measures total lean soft tissue, which includes the connective tissue and other non-functional components. And so people are now beginning to find deuterated creatine gives better measures of outcome. This just shows the creatine dilution method prediction of muscle mass against MRI measurements of muscle mass, and they're highly correlated. But you'll see more and more of this in the literature. Now, what we find then, I'll take this one step further. I'm sure you all know what bioimpedance is shown here. And bioimpedance can give estimates of extracellular and intracellular fluid and a measure called phase angle, which also is a derivative of this technique based on resistance and reactance. And Yamada in Japan, or actually here in the US at the time, looked at the extracellular to intracellular water ratio in the legs as a function of strength. And the reason this is relevant is that as people get older and they have more muscle atrophy, the extracellular space increases and the intracellular space shrinks. The muscle contractile fibers shrink. And he found then that that was associated with muscle strength. The more atrophy you had of your muscle, the weaker they were, and the more extracellular water was present. Now, it's a little complicated slide, but I'll take it one step further. If you measure, this is a study done by Brianna Bourgeois a couple of years ago. She had a large sample of adults, and she looked at lean soft tissue mass measured by DEXA versus age. And as you can see from the regression line, there's a very low slope, very little change across the ages. But she found in the same people that their strength decreased significantly across the ages, again reflecting that phenomenon that strength proportionally decreases more than mass as measured by DEXA. But also what she found is with BIA, there was a decrease in phase angle and an increase in echogenicity by ultrasound, reflecting more connective tissue and fat as people got older. And what she found is that if she combined a phase angle measurement with BIA with the DEXA measurement of lean soft tissue mass, she improved the prediction of strength. What that reflects then is that we can capture mass by DEXA and quality of the muscle by BIA or echogenicity or and what this is telling us is that combining these methods then can improve our understanding of what these tissues are made out of and their functionality. Now, one of the great advances as I started out my presentation is the understanding that we all differ in body shape. In this very wonderful study by Steph in 2017, he found that when he measured shape using here MRI in people, he found in a large sample of adults, he found that there was a phenotype that had low leg fat mass, fatty liver, visceral fat, even if they were normal weight. And he coined this idea, the metabolically unhealthy phenotype, and that this might reflect some type of lipodystrophy, that is the low fat mass in the legs. And as some of you may know, there are genetic syndromes, lipodystrophy, where people can't make normal adipose tissue. And the fat then travels to the liver. People have very high insulin resistance, fatty liver, and diabetes. And they conceived of this as a type of lipodystrophy, but not common, at least in their view. But nevertheless, it suggests that people are walking around with this phenotype. And I'm going to show that, in fact, they are. It's quite a common phenotype. And to begin that journey, then, I'd like to talk about how do we measure shape in people beyond the composition? How do we quantify variation in body shape? And this work really began in earnest in Loughborough in 1989. They were looking for a way to fit people better for sweaters made by the garment industry there. And they developed a photogrammetry technique. It's called photogrammetry, using various cameras and scanners. And that morphed very quickly over the last few decades to laser scanners shown here, where you could then put a person in a laser scanner device like the one shown here, a couple of seconds, and then you can scan the person, and that then gives you a detailed map of their body shape. And these are quite expensive. The devices are more than $100,000. The software alone is very expensive, $80,000. But that's given away with the introduction of the Microsoft Kinect using the gaming industry to build what I'm showing here, these three low-cost devices. These are in the $5,000 to $10,000 or $20,000 range, and the left a little more expensive. But these devices have low-cost cameras in them, and they are different technological designs. The one on the left has multiple cameras. The two on the right have a rotating platform and a single camera. And from these devices, then, using various infrared sensors or time of flight technology and very simple algorithms, you can quantify somebody's whole body avatar detailed map and their body shape, just shown here. And then you can use software to measure various body circumferences, just a few of examples shown here. And now these devices can give you 300 or 400 different body circumference, length, volume, surface area, and other estimates. And of course, the more you pay, probably the higher the quality will be, but that's changing very fast. So this is changing the anthropometry world dramatically. No tape measures are needed. And this just shows a few examples of validation studies. These devices generally tend to validate quite well. They may not give you the exact waist circumference that is the standard one. They have their own proprietary measurements. But generally speaking, the correlation between tape measure and 3D optical measure is quite strong. And these devices, even as I speak, they're changing. Here's one you can put in your home if you're interested in your body shape changes on a day-to-day basis. This one's under $1,000 called Naked Fitness. And we looked at in our lab, worked perfectly compared to tape. It gave the right waist, the right hip, various arm circumferences, and so on. And the software in these are very elegant, and these devices are changing quickly. And you can do longitudinal studies, of course. This woman lost 5 or 10 pounds over a month or month and a half interval. And you can see the software then allows you to generate the silhouettes, and just shown over here, the changes in the waist and hip at the two different time points. So you can quantify change in shape over time. And the conventional way this works, you scan the subject as shown on the left, that creates a software file. And that file then is used to landmark to measure the dimensions you're interested in, the volumes and circumferences. And in turn, you can build models then that will predict blood lipid levels, body composition, and ultimately health risk. That's the upper part. More advanced, though, approaches is you can put what's called fiducial points on a person, and you do those by hand, and then that allows you to fit a mesh to the person, a standardized mesh, and use mathematical techniques such as principal components analysis to get detailed body shape indices. And then again, to use that information to predict health risk. But it also can predict something else, which is you can predict what someone's DEXA scan would look like or what their MRI scan would look like by doing principal components analysis relating your shape to a large library of scans that are from MRI or DEXA. I'm going to show you a few examples quickly, but this just shows these various fiducial points that used to be placed by hand, but now it can be done automatic. And here's a little software program my colleague Brian Corliss, University of Washington in Seattle, built. What this does, shown on the left, you have a little slider, and you can measure, move that slider, change the person's height, their weight, their limb circumferences, their body circumferences, and so on. You can slide it. So here's the standard baseline person. And then these principal component shape models allow you to vary their weight. So here I slide it up a little bit in their weight, and this is what the person would look like at the higher weight. This software is becoming more widely available to do this, and it's based on real bodies and principal component analysis. And here's my colleague, John Shepard. In the middle, his BMI is 28. And we built the software then to vary fat and lean independently. So John said, what would I look like if I lost 20 kilograms of fat on the left? And what would I look like if I gained 20 kilograms of fat on the right? And then he said, what if I change it to add 20 kilograms of lean to that? And you can see the different phenotypes John takes on then. And so we can build these models then using the libraries of scans we have to quantify these variation in shape. And this just shows one example. And I think we can do better now than this is a little older. But we can then do a 3D optical scan on you and predict your DEXA shown on the right and the actual DEXA on the left. In the middle, the difference, that's a man. And here's a woman DEXA. And we're pretty good, very close. It's not a real scan. This is just a proof of concept, but a very fascinating type of analysis you can do with this type of imaging technology. And most of the scanners I've shown are very small. Here's one called the Dome that we have at LSU. We built one, my colleagues at LSU built one. This one, they can do the whole football team inside this little green mat. Basically, they line them all up, they throw passes, they do all kinds of maneuvers, and they get the 3D avatars. And then they can build games and other activities then from the actual players using their 3D optical image. And we're using this to scan little kids. This just shows an avatar of a mannequin, a little baby, young child mannequin, but we can make very large optical devices like this, and we can do studies in very hard to evaluate young children like that. Also, this technology, as I'm sure you know, is going very quickly to handhelds. So for example, these are green screen images we use to capture data on individuals. And then we can build these into software. This just shows a typical handheld device where you stand in front of it, you get your image, and then that in turn does the analysis and gives you your body fat. These are becoming widely available, and it's the same idea. And this one's the most recent one we studied in our lab. This is the Halo. Amazon makes this, and you can use this coupled with your cell phone to capture images. The device then creates a 3D avatar and gives you a quantification of your body fat. You can use these avatars then to measure circumferences. Amazon hasn't done that yet, but nevertheless, you can see the direction this technology is going. And if you want to play around with this, you can buy this one for $150 from SizeStream. It comes with a little outfit and a rod and a little mat you stand on. Someone walks around you with the cell phone, takes your images, and you get a beautiful avatar from which you can get many body dimensions, the lengths, circumferences of your body, and so on. Now, why does this all matter? Well, here's a little study we did going back to that idea of the lipodystrophy. John Schoen and I did this together not too long ago. We took people, men in this case, but women also from NHANES, and we divided them into tertiles based on their adjusted waist circumference. In this example is a normal weight man, and this is what the man would look like if he had a low waist circumference. This is the identical man, same height, weight, and age, with a high waist circumference. And you can immediately see the difference in the phenotype. And the red area is the difference between the two men generated by principal component analysis of their optical scan. And what you then find is when you go into the phenotype of these individuals, this is roughly 1/3 of the population looks like the man on the right, and 1/3 looks like the man on the left, the rest being in between. It's a very common phenotype. And what you find is that the man on the right will have higher body fat less muscle mass and bone, more trunk fat, and more leg fat than the man on the left. The same applies for women. In other words, this phenotype, very simply defined by a high waist circumference, even after adjusting for weight, height, age, and race, defines a very distinct phenotype that is more than just visceral fat, as some may say. And where is this all going then? Well, in this particular area, what's happening is many labs around the world are generating 3D optical data shown in the bottom using these various devices and DEXA. And these in turn, this data then can be loaded up into a cloud storage area, and assuming you agree to it. And then you can, ultimately, there can be millions of studies within this universal body shape composition database. And then it's coupled with machine learning software that then will generate body composition equations, energy expenditure, and risk prediction models for users. And so this is an iterative process. It's ongoing. The more data is loaded, the more specific these models become. And if you think back about the simplicity of the Harris-Benedict equation for predicting resting energy expenditure, or equations like that, that's going to be a thing of the past. As we get better and better at building these global databases with this high-level technology and the coupling of machine learning, artificial intelligence, will radically transform our ability to study metabolism and body composition. And we're not alone in thinking about this, because people like John Ioannidis at Stanford and others, there's a lot being written on the idea that our archaic approach to developing prediction models and clinical modeling of every center has their own specific, there's the Harris-Benedict, the millions of equations for predicting resting energy expenditure. That needs to give way to much more concerted global efforts to building these kinds of models using multiple centers, standardized technology, and so on. So I've traveled through a lot of different areas. I've hoped to give you a little bit of an idea of some of the evolving concepts, and I can't help but thank my many colleagues who have helped me travel on this journey. Thanks very much, and thanks for inviting me, John.
All right. Give me 1 second, Steve. Can you stop the share screen on your end for just a second?
Yep.
All right. Thank you, Steve, for the presentation. As I mentioned to the audience before, if you have questions, please direct them to me. We do have about 10 minutes or so left. There are a couple of questions that I'll be asking for, or that I have already that I'll be asking. But if there are any more, please send those to me in the chat, and I will try to work them in time available to us. So let's see. The first question that I have, you mentioned how the BOD-POD is the only available air displacement plethysmograph available for measuring total body volume, but that there were others in the past. In regard to these other air displacement plethysmographs, were these kind of one-off devices built in labs, or were these commercially available ones that you could buy?
John, that's such a good question, because I just wrote a little review on measuring body volume, and I found 7 or 8 different types of devices people have tried. And there are various, you can build these chambers and inject, for example, xenon or helium or various chemical compounds and get a pretty good estimate of volume. And I would say that if somebody was interested in it, they could devote some time and probably figure out a better way of developing body volume estimates than BOD-POD alone, which is a fine technique. The earliest BOD-POD devices were built 50, 60 years ago. They weren't very accurate. There were a lot of technology problems, but BOD-POD really invested a lot in solving them. But nevertheless, I would say if that's a topic someone's interested in, there are a surprisingly large number of techniques people have tried over the years.
Okay, thank you. Next question. Has there been any epidemiological work looking at health risks associated with the very specific adipose or fat tissue depots that you mentioned in terms of their health risks, so we can get a better handle on how to use that imaging going forward?
Yeah, there's quite a large literature on ectopic fat depots, and they tend to be intercorrelated. For example, if you have a lot of visceral adipose tissue, you tend to have a lot of myocardial lipid uh, and pancreatic lipid. So they're not completely independent, but nevertheless, there's a lot of literature now on looking at various outcomes, starting intermediate outcomes like insulin resistance all the way up to diabetes risk and things like that. Yeah, surprisingly large literature, might be worth someone sitting down attempting to review it. I didn't find any reviews, but it definitely is there as individual papers.
All right, thank you. Next question. If we measure the absolute masses of all the body's different tissues and then multiply them by their tissue-specific metabolic rates, which are typically determined in vitro, how does that estimate of resting energy expenditure compare to what we might be able to estimate via indirect calorimetry or direct calorimetry?
It's a great question. Right now, the models we have, which are based on 6 or 7 components like adipose tissue, muscle, heart, liver, kidney, brain, things like that, they come up certainly within plus or minus 5% in an individual person. Uh, Really, it's quite fascinating that it can be so accurate in this sense because you're measuring a lot of different things. But I think the great barrier we have in studying metabolism now, or certainly resting metabolic rate, is we can't measure metabolic rates in vivo of individual organs other than with invasive methods. And so I think if I had another career or two, I would focus on trying to figure out how to do that. We can measure ATP turnover and so on, but obviously the numbers I showed are just averages. And I know we don't have a lot of time, but I've given a lot of thought to an organ like the heart. And now that I measure my heart rate, with my little devices 24 hours, you see how amazingly variable your heart rate is. Of course, heart rate times blood pressure is your heart metabolic rate, myocardial oxygen consumption. And so our heart energy expenditure is varying over 24 hours. So it's not really a constant.
Okay. Um, Next question. How do laser scanner estimates of body volume compare to those obtained from the more traditional underwater weighing or via air displacement plethysmography? And is there dramatic differences between the laser scanners and the video game imaging methods that you showed?
Yeah. Yeah. Oh, such a good question. The answer is that let's take BOD as an example. It doesn't get gas in the GI tract. You have to make an assumption about it, and you have to measure residual volume. The laser scanning just measures your exterior. So it has all of those things still in there, right?
Yeah.
Gas in the bell. So they don't match up perfectly, but the good quality laser scanners get very accurate surface volumes. You still have to get the other volumes. Uh, Now, the other devices also correlate highly with things like BodPod, even down to the $1,000 version devices or less. So they're very good at getting volume. But here's the catch. To get an accurate body fat from volume, you need a very, very accurate volume. And being plus or minus a liter is probably not good enough, or plus or minus 2 liters. So what you find is using the volume estimates from these devices really is not a substitute for getting body fat.
Okay.
Yeah.
And then with time, I think we have time for just one more question.
Yeah.
With being able to review MRI and test for D-creatine in urine samples, how likely is it that we could start having clients where the regular doctors run these tests to get a more accurate evaluation of body composition?
Another great question. There was a company that I don't, maybe others know more about it than I do, rented MRI scanners when they weren't in use, said you can come in at 7 p.m. on Sunday and get a whole body MRI scan for $50, and then we'll analyze it for another $50. So that potential is there. And what I'm seeing on the horizon is more and more engineers developing automated softwares for MRI, including organ volumes. And I think given 5 years, you're going to see the cost of these things coming way down. And that would be a huge breakthrough because now our MRI databases, whole body databases, scanned in the hundreds. But if we got up like DEXA, where you did 40,000 DEXA scans and then Haines, something like that, you see things you cannot possibly see in smaller samples.
All right. Well, thank you, Steve. It's time for us to conclude. I want to make an announcement though before you go. We do have 2 concurrent sessions which follow this at 10 a.m. Pacific, 11 a.m. Mountain. Those being the Curriculum Vitae workshop with Cheryl Minnick, and then the open forum professionals, how you doing, with Dr. Lex Gidley.